Hello again, this is Earl Silverman, Editor-in-Chief of the Journal of Rheumatology. Welcome you back as I highlight a paper of particular interest to the readership by interviewing three of the authors of an article from the December 2022 edition of the journal. The article I picked for this more in-depth exposure is entitled Immune Response to SARS-CoV-2 Third Vaccine in Patients with Rheumatoid Arthritis who had no seroconversion after primary two-dose regimen with inactivated or vector-based, by Dr. Carolina Isnardi and colleagues. Today, I'm pleased to be speaking to Drs. Carolina Isnardi, Guillermo Pons-Estel, and Gustavo Cerda about their paper about immune responses to the third SARS-CoV-2 vaccination. I want to thank all three of you for taking the time to speak to me and our listeners. So let us begin.
Hi, good morning. Thank you for your introduction and the invitation to participate in this interview. So we are glad to be here.
I'm glad you could make it. In your article, you focus on the 21 patients with RA who did not respond to the first two COVID-19 vaccination doses. Can you please let the listeners know what the total RA cohort who were vaccinated that you chose these 21 from, and how many of these patients did have antibody levels tested?
Great. This is actually a two-step study. In the first phase, 120 patients with rheumatoid arthritis were included. They were vaccinated for COVID-19 according to our national plan, meaning they received all types of vaccines, inactivated mRNA vaccines and viral platforms, and also homologous and mixed regimens. The humoral and cellular response were assessed for after the first and second doses, and after the second dose, 82% of them presented anti-spike antibodies, 70% neutralizing activity, and 65% T-cell response. And for this second phase, the one we are reporting in our manuscript, those who did not develop anti-SARS-CoV-2 antibodies were recruited.
Okay, thank you. Can you please review the major findings of your study?
Can I ask a comment?
I'm sorry, my bad.
Sorry, no, it's okay. But it's, the idea was not only a scientific interest, but also an ethical consideration, because, in those patients not having antibodies or not having neutralizing antibodies, they felt vulnerable. So we really felt that we had to offer these people something to have a protection against COVID. So and that was the reason that we talked with the authorities and we offered them after those.
Oh, thank you. And just, when was the study undertaken? When did it finish?
Okay, we started recruiting patients in April 2021. They received in that period the first dose, and during June and July 2021 they received the second dose. Okay. For the third dose, we recruit patients between November and December 2021, and that's when we assessed we got the blood samples and we assessed the humoral and cellular response.
Okay. And can you please review the major findings of your study?
Yes, of course. During the second phase of the study, the one when we assessed the third dose efficacy, the selected patients received a third dose of vaccines, most of them RNA vaccine, one of them a vector-based vaccine, and again, development of anti-spike IgG neutralizing activity and T cell response was assessed between 21 and 40 days after the third dose. And after this vaccination, 90% of the patients presented detectable anti-SARS-CoV-2 IgG, and 76% of them neutralizing activity. Compared to the treatments, abatacept and rituximab were associated with the absence of neutralizing activity and lower titers of neutralizing antibodies, and specific T cell response was detected in 71% of the patients selected after the third dose. And in that case, the use of abatacept was associated with a significantly lower frequency of T cell response. And that's what those were our, that's what we find out, and that's what we wanted to highlight at this point.
Any additional comments?
Yes, I want to add here that this was a joint venture, as Gustavo said before, between the government of the Provincia de Buenos Aires, the research unit of the Argentine Society of Rheumatology, and two centers in Buenos Aires. And this is a unique, in some way, a unique study, because most of these patients at this stage in Argentina were vaccinated with Sputnik 5. So we got from the government the possibility to use the Pfizer vaccine. So probably there are not many places around the world that can have this combination of vaccine showing the results that Carolina are describing.
At that point, all of the patients, all 21 patients, had mixed regimens, because they could have the same dose between the first and the second, but then when they got the third dose, they all got at least two different vaccines, two different types of vaccines, I mean.
And just for my clarification, it sounds like it was approximately 6 to 8 months after their second dose. You gave them the third dose in that ballpark?
After between 4 and 5 months.
4 to 5 months,
dose.
okay. And it is interesting because, you know, a lot of the studies have shown what the good response even after the second dose if you switch vaccines. So if you have either the vector or inactivated and you switch to the mRNA, even the flip seems to give you a better response than if you stay with the same vaccine. And that certainly is, who knows, but that whether it's a third dose because you don't have a control, or whether it's changing to the mRNA, but it's also certainly intriguing the excellent response you had. So for somebody like me who doesn't know what a mean level of neutralizing antibody of 12 means, I assume that's protective.
Yeah.
Yes, exactly. I mean, we know that neutralizing antibodies are very important to control SARS-CoV-2 infection, but unfortunately, there is no agreement about a definition of protective neutralizing antibody titer. I mean, there is no cutoff value. Additionally, different studies use different assays to assess neutralizing activity, and that make it difficult to compare between studies. And particularly in our study, the neutralizing titer was calculated as the inverse of the highest plasma dilution, showing 80% inhibition of the cytopathic effect. And as you mentioned, we observed that our cohort median neutralizing antibodies titer was 1 in 12, which is similar for what clinical trials showed. But on the other hand, I want to highlight a very interesting study from Australia, which analyzed data from multiple vaccine trials, showing that the estimated 50% protective neutralization level from infection was 20% of the mean convalescent level, which is a normalized value they use to compare the results. And this is approximately between 1 in 10 and 1 in 30. That means that our results are in the middle of this range. And while the neutralization level for 50% of protection from severe infection was 3% of the mean convalescent level, which is significantly lower than this 20% level required for protection from any symptomatic infection. So there is no cutoff point, but using these results, we can say that our results, 1 in 12, should be protective.
Thank you. Any further comments?
The other factor that could affect the results and the response in our patients was the fact that even when the ACR recommendations regarding stopping DMARDs or biologic DMARDs were published at that time, due to the vaccination scheduling in our country, sometimes patients receive the appointment one day before, and we didn't have time to stop methotrexate or biologic DMARDs with the proper period of time. So that also could affect the response in some patients.
That's very interesting. That makes you want, it's still a good percentage of patients with neutralizing antibodies.
Yeah.
It makes you wonder about the recommendation, certainly for some DMARDs, is their half-life is long. So I always wonder about it. So, no, unlike methotrexate, but certainly some others. So one of the intriguing things, as a trained T-cell immunologist, you found that 41% of patients had a T-cell response after the second vaccination, despite no antibody response. So is your feeling about the relative importance of the T-cell response vis-a-vis, regarding protection in the absence of a B-cell response?
Well, we know that in general, antiviral immune activity is carried out by both neutralizing antibodies and cytotoxic response, which aims to eliminate intra and extracellular viruses from the body, meaning that B and T cells are important to facilitate SARS-CoV-2 elimination. But so when we assess the specific T cell response, we are evaluating the ability to carry out the cytotoxic response and destroy infected cells. We usually assess the humoral response because it's easier and it's cheaper. So that's why globally the humoral response is more often assessed. And we know that, as I had mentioned before, that the neutralizing activity is very important to control the infection. But T cells are also important. And for us rheumatologists, it's very important to know that some patients who did not develop antibodies after vaccination could still perform, not all, but some of them could still perform a T cell response. This is particularly important in those taking drugs which interfere with B cells functions like rituximab. In our study, none of the 4 patients receiving rituximab developed neutralizing antibodies after 2 doses, and 2 of them developed or had a positive result after 3 doses. But all of them had a positive T cell response. And this is similar as what other studies have already mentioned, that those patients develop at least part of the expected response after vaccination. And we know that we have to wait as long as possible between the last rituximab infusion and the vaccination, but that's not always possible. And that's at least something we can offer the patient, and we have to vaccinate it no matter what.
Any other comments?
Yeah, this is very interesting, and there is also an interesting letter in Annals of Rheumatic Diseases coming from the Austrian group. It's only in patients receiving rituximab. I think in 6 patients or something like that. Correct me, Caro, but they observe what Carolina has mentioned, that those patients not having a B cell response had a T cell response. So it's very important to say our patients that even when they don't develop antibodies, they can have a T cell response.
It's very interesting that 2 things enter my mind about that. One, did you, prior to looking at the antibody response in the rituximab patients, have a chance to look by flow cytometry at the repopulation of the B cell compartment? Or, I mean, it's only 4 patients, but you said half of them had a antibody response after the 3rd dose. Did you measure their B cell numbers, like with CD19?
No, in that case, we were not able to
Just
do a
curious.
cytometry, flow cytometry or something.
But
like
I think, but I think that the 3rd dose was after what, you know, what period of the last dose?
Around
months.
Around 14 months.
Yeah.
Okay, so it is supposed that the BCR response was restored after 11 months. Yeah.
Yeah, most of them would be populated. I agree. So the other thing that's intriguing about the T cell response, again, is that certainly clinically, people receiving rituximab don't seem to be overly infectable. They don't seem to have a significant increase of viral infection. Coming back to the potential importance of the T cell response, even the absence of antibodies. So it just, do you have any data on, I know this is a hard question, people who had a T cell response despite no antibody response versus those who didn't have both regarding infection? I know it's small numbers. Did you see many infections, I guess, in RA patients is my real question.
Well, right now we are following up these 120 patients, and we are planning to publish the evolution of these patients during the 1st year after vaccination. And we are still collecting data, but we already know about these 4 patients with rituximab. And unfortunately, one of them not having humoral response, but T cell response, he died due to COVID-19, a severe COVID-19 infection. So that's the only data we have right now about this group of patients. But we have data from another cohort of patients we are collecting in our country. This is the SARS-CoV registry, and we saw that patients, some of them were vaccinated, some of them not at the time of the infection, but those receiving rituximab, similar what this was published by our courts, had 7 times more, were 7 times more likely to have be hospitalized or have a poor prognosis of the infection. So this is something we have to have in mind in these patients with rituximab. They have poor prognosis, and they also have a lower response after vaccination. So there's a group of patients we have to remind every time we decide to use rituximab in this context.
Something else that we may be able to add in the near future is that, as Carolina said, we are following this patient. And in Argentina, we have a regimen of vaccination that goes from 4 to 5 doses of vaccine up to date in this present time. So to see what happened with those patients that were treated with rituximab over time may give us the chance to see what is the clinical response of these patients, the levels of infections, and this is something that may be added by the follow-up of this cohort.
It's fascinating. And just out of my curiosity, are you now routinely giving these patients the mRNA, or is it still a mixture of vaccinations in Argentina?
Still a mixture. But we have more mRNA now. Right. We have Pfizer and Moderna.
Okay. So along those same lines, it was interesting, especially we talked about rituximab, but your findings with abatacept, who didn't seem in your study didn't seem to respond either in the B cell nor the T cell compartment. Do you have any data? You were commenting a little about infections in the rituximab group. How many patients were treated with abatacept, and any clinical follow-up on that, out of curiosity?
In the first phase, 11 patients with abatacept were included, and if I'm not wrong, 6 of them were included in the second phase. And we know that abatacept affects not only the B compartment, but also the T cell activation. So in this context, we saw that the patients taking abatacept were associated with a deficient humoral and cellular response. And even after the third dose, 33% of the patients and 66% of the patients did not develop a neutralizing activity nor a T cell response, respectively. And 2 of them, they did not develop neither humoral nor T cell response. Unfortunately, I don't have data, clinical data about the evolution of these patients. We are following them up, but I don't have the data yet. But using data from other studies, we know that these results are comparable with those, and that patients, they seems to perform better than those taking rituximab, but there's not conclusive data about that.
Okay. Any other comments?
No, but it's hard to compare. Not in our study, no, but in other studies, it's hard to compare because, patients receiving abatacept are patients with array, and patients receiving rituximab are a mixture of patients with array, with vasculitis, with lupus, with other diseases. So it's hard to compare that data, no?
Yeah, absolutely. We'd have to just keep it to RA, presumably.
Yeah.
The numbers become small.
Yeah.
No, thank you. Excellent. Do you think, is there anything else you'd like to add that we have missed and you think the listeners should know?
Well, I would like to highlight the importance of vaccination. Even though we include a small sample of patients during both phases of our study, we were able to show that 70% of the patients who did not seroconvert after 2 doses of vaccines achieved neutralizing activity after the 3rd dose. And at this point, we know that vaccines are safe and they have allowed to reduce viral spread and cases of severe COVID. So I think it's our job as physicians to encourage of patients to get vaccinated with complete regimens, and including all additional doses, like Guillermo just mentioned. We now have to get our patients to get the 5th dose of vaccine. So that's our job, and data now available highlights the importance of this tool.
Yeah, I agree. I also will add that I think that it's important also to measure these antibodies or their response in the general population, and mainly in patients with rheumatic diseases, because we don't know now what we have to do or how to follow our patients if they need a 5th dose of the vaccine, how often they have to be vaccinated. So I think that it's very important to collect data on the response and the duration of the response.
I totally agree with Gustavo. We see every other day in the console that we found patients that ask us if they have to give themselves the 3rd, the 4th, or the 5th dose. And I think that an important point to highlight in those patients, if we ask for the titers of antibodies and those are low, this is something important that we can explain the patients an easy way to explain the patients that they are not covered and they are in a high risk of developing COVID. So this is something important that all the listeners, I think, should take as a home message.
I agree completely. Certainly in Canada, it is not, titers are not routinely available commercially, only under special circumstances, and our government currently does not consider autoimmune diseases on immunosuppressive medications a reason to look at antibody titers against a spike protein. Is that similar in Argentina? Is it only done in research labs under studies like such as your study?
Yes, absolutely the same.
Yeah, I certainly hope that the community would get the regulatory agencies to change that policy, because it would be very helpful in convincing patients who are sitting on the fence whether they should get
Yes, agreed.
repeated vaccination. Okay, anything else you would wish to add?
Just thank you very much for this opportunity.
Well,
It has been a pleasure to be here.
Exactly. Thank you.
okay, and I want to thank you, all three of you. It was great for taking the time for our listeners about really the importance of, at this point, the third dose for patients with RA, but now, as you correctly pointed out, in many countries, if not all, hopefully in all countries, fourth and fifth doses are now available. And I hope that our listeners will please read the entire manuscript, Immune Responses to SARS-CoV-2 Third Vaccine in patients with rheumatoid arthritis who had no seroconversion after the primary 2-dose regimen with inactivated or vector-based vaccines by Dr. Carolina Isnardi and colleagues, and as well as please read the accompanying editorial entitled "The Third Dose is a Charm, Effective Cellular and Humoral Immune Responses to Third COVID-19 Doses in Immunocompromised Non-Responders" by Drs. Wilson, Cuswanto, and Matthew Baker. Both of these are available at the journal's website at jroom.org. And I'd love to hear any comments or questions, either via Twitter at jroom or by email at manuscripts@jroom.com. And I want to thank my guests as well as the audience for joining us, and hope you'll join next week. Next month. Maybe I'll be doing it next week, but you won't hear it next week, when I speak to the author or authors of one of the articles of particular interest that I selected for the January 2023 edition. And I wish you all happy holidays and to a good new year. Thank you.
Thank you.
Thank you. Bye-bye.
Thank you.